In the mini-series here, we've spent a lot of time discussing bacterial infections, and in this short segment, we're going to shift our attention to viruses and ask a broader question. How do important viral pathogens enter the body, and what defenses become most important once they do? This segment serves as a bridge between microbiology and immunology. We'll start with organizing viral pathogens by portal of entry, such as respiratory tract, GI tract, skin, genital, and blood. And from there, we'll connect those entry routes to the body's non-specific defenses and adaptive immune responses. The goal here is not to memorize every virus on a list. The goal is to think clinically. If we understand how a virus enters the body, we can better understand what symptoms may appear, what defenses are involved, and how exposure history helps us narrow the differential diagnosis. This slide is meant to be an overview or summary, not a list to memorize line by line. The important idea is that viral diseases can be organized by portal of entry. That framework helps us think clinically. If a patient presents with respiratory symptoms, we start thinking about viruses that commonly enter through the respiratory tract, such as influenza, rhinovirus, RSV, or Epstein-Barr virus. If the symptoms are gastrointestinal, then viruses such as hepatitis A or rotavirus may move higher on the list. If there is a skin exposure, bite, or lesion, then we think differently. Rabies, HPV, and pox-related infections become more relevant. If the route involves genital contact or blood exposure, then viruses like HPV, hepatitis B, hepatitis C, HIV, and cytomegalovirus become more important considerations. So rather than memorizing every virus and disease pairing, I want you to focus on the pattern. The route of exposure helps narrow the differential diagnosis. In other words, where the virus enters the body often gives us important clues about what disease process might follow. This is also a useful reminder that viruses are not all spread the same way. Some are mainly respiratory, some are gastrointestinal, some require skin or mucosal contact, and others are transmitted through blood or sexual exposure. That matters for prevention, patient education, and clinical decision making. The takeaway from this slide is simple. When evaluating a possible viral illness, do not start only with the name of the virus. Start with the exposure history. Ask how the patient may have encountered the pathogen, what body system is involved, and whether the route of entry fits the symptoms you are seeing. In this slide, we are focusing on the body's nonspecific defenses against viral infection. These are the immediate, broad protective mechanisms that do not require the immune system to recognize one specific virus in advance. Instead, they help limit viral replication, clear infected material, and make the host environment less favorable for the virus. One important example is the interferon response. Interferons are cytokines released by infected cells and nearby immune cells. Their job is to warn neighboring cells and help limit viral replication. More specifically, interferons can inhibit viral translation, which means they reduce the ability of the infected cell to produce viral proteins. Since viruses depend on host cells to make more copies of themselves, anything that interferes with that process can slow down the infection. Natural killer cells are another major nonspecific defense. Recall from earlier in the lecture that NK cells are especially important when a cell is not displaying MHC1 properly. Healthy cells generally display MHC1 on their surface as a way of showing the immune system, I belong here. Some virus-infected cells reduce MHC1 expression as a way of hiding from CD8 T cells. Natural killer cells help close that loophole by targeting cells that fail to display MHC1 appropriately. Phagocytosis also contributes to nonspecific defense. Phagocytic cells help clear pathogens, infected debris, and damaged material from tissues. This does not necessarily mean they are specifically targeting one unique virus. Rather, they are helping clean up the battlefield and reduce the burden of infectious material. The body also uses antiviral peptides and enzymes. Defensins are small peptides that have antiviral and antibacterial properties. Another example listed here is the apolipoprotein bRNA editing enzyme, which can affect viral nucleic acids. The details of that mechanism are more advanced than we need to emphasize here, but the key point is that the body has built-in molecular defenses that can interfere with viral survival and replication. Fever is another important nonspecific response. We often think of fever as a symptom that makes the patient uncomfortable, but fever can also be protective. Many organisms do not replicate as effectively when body temperature rises. So while fever can become dangerous if it is too high or prolonged, a moderate fever is not simply a problem. It is part of the body's attempt to create a less favorable environment for pathogen replication. Mucociliary clearance is especially important in the respiratory tract. You can think of this as the mucociliary escalator. Mucus traps particles, pathogens, and debris, while cilia help move that material upward and out of the lungs. This is one reason respiratory tract health matters so much. If mucus production, ciliary movement, or airway clearance is impaired, the body loses an important mechanical defense. Finally, we need to remember that host defenses are modified by the condition of the host. Age matters. Infants and older adults may not respond as robustly, and vaccine responses can change across the lifespan. Stress also matters because chronic stress can increase cortisol, and elevated cortisol can suppress aspects of immune function. Nutrition matters as well. If a patient is malnourished or underfed, they may not have the resources needed to build and sustain an effective immune response. So, the practical reminder from this slide is that viral defense depends on both pathogen control and host condition. The same virus may not affect every patient in the same way. A well-rested, well-nourished person may handle an exposure differently than someone who is sleep-deprived, chronically stressed, underfed, or physiologically strained. That is why clinical context matters when we think about infection risk and immune defense. For this slide, we are moving from nonspecific defenses back into the adaptive immune system. These are the defenses that depend on antigen recognition, immune coordination, and memory. Instead of responding broadly to infection, the body is now responding to something specific. The first major player is the CD8 cell. Remember, CD8 cells are killer T cells. Their job is to identify infected or abnormal cells and trigger those cells to die. This is especially important in viral infections because viruses replicate inside host cells. Once a virus is inside the cell, the immune system often has to eliminate the infected cell itself to stop the virus from making more copies. This is where MHC1 matters. Almost all nucleated cells display MHC1 on their surface. MHC1 acts like a display platform. If a cell is infected, pieces of viral protein can be presented on MHC1. When a CD8 cell recognizes a matching antigen on MHC1, it interprets that as evidence that something abnormal is happening inside the cell. The CD8 cell then causes that infected cell to undergo cell death. That sounds destructive, but it is protective because it helps limit viral replication and spread. Next, we have CD4 cells. CD4 cells are helper T cells. Their role is not primarily to kill infected cells directly. Instead, they help coordinate the immune response by secreting cytokines. These cytokines support many other immune cells, including B cells, macrophages, and CD8 cells. So if CD8 cells are the search and destroy cells, CD4 cells are more like command and control. This is why CD4 cells are so clinically important. Without functioning CD4 cells, the immune system becomes much less effective at coordinating defense against infection and cancer. This is one reason HIV is so serious. HIV targets CD4 cells and can gradually deplete them over time, leaving the patient vulnerable to infections and malignancies that the immune system would normally help control. Antibodies are another major part of specific defense. Antibodies are produced by B cells and bind specific antigens. In viral defense, antibodies can help neutralize viruses before they enter host cells. Once a virus is already inside a cell, CD8 cells become especially important. But before entry occurs, antibodies can bind viral particles and reduce their ability to infect new cells. Antibodies also support adaptive memory, which helps explain why vaccination or prior infection can lead to a faster response later. Passive immunity is a little different. Passive immunity means antibodies are given to a person rather than produced by that person's own immune system. This can happen naturally, such as IgG crossing the placenta before birth or IgA antibodies being transferred through breast milk. It can also happen medically through immune globulin products. For example, after certain serious exposures, such as rabies or tetanus risk, a patient may receive immune globulin for immediate antibody protection. The advantage is that passive immunity works quickly. The limitation is that it is temporary and does not create long-term immune memory. Finally, herd immunity is the population-level extension of adaptive protection. If enough people in a community are immune, either through vaccination or prior infection, transmission chains are interrupted, the pathogen has a harder time finding susceptible people to infect. This also helps protect people who cannot respond well to vaccines, including some immunocompromised individuals, newborns, and others with weaker protection. The key takeaway is that viral disease is not just about the virus. It is also about route of entry, host defenses, immune memory, and population protection. CD8 cells kill infected cells. CD4 cells coordinate the response. Antibodies help neutralize pathogens. Passive immunity can provide temporary protection, and herd immunity helps interrupt transmission at the community level.